Great. Well, so we've spent an awful lot of time thinking about sequencers, sequencing this, sequencing that. Maybe it's a transcriptome, maybe it's a genome. And a lot of people in bioinformatics are content to play in that field. They've spent a lot of time thinking about sequence data and how to make the best use of them. However, I've spent the last 20 years of my career working in the field of proteomics. That gives me a great pleasure because I get to talk about that field today. I just get that one day, and then on Monday we'll be talking about quantitation in proteomics and metabolomics. And then we have a quick run at image processing and flow cytometry. And finally, a quick look at biological pathways and networks. So we're all going to be finished with this on Wednesday afternoon. And then I'm going to make an arrangement with the molecular physiology folks that you get to take the quiz on a day after that, whether it's the following day or not, I don't know. But it'll be the same format as all of these, so it shouldn't be anything too unfamiliar. So with that said, let's dive into the fine world of proteomics, or as I think of it, protein identification, and protein structure, the other part of this. Now, let's move ahead. And so we're going to be spending a lot of time talking about tandem mass spectrometry. Stellenbosch University is one of a few universities within South Africa that have invested significant money in mass spectrometry instrumentation that allows us to identify proteins. So these are tandem mass spectrometers. We will talk a little bit about how these instruments work, because after all, if you don't understand where the data come from, you probably won't understand what you can do with them. So we will spend a little time on how this technology works, move then into sort of the canonical bioinformatics pipeline for making sense of these data, which is the database search algorithm. At last, we'll talk a little bit about how we figure out whether those algorithms worked or not, and how we build back a protein list from those peptides. And with that, we will move into the fine world of protein structure. Now, protein structure is a part of this lecture that I decided to rebuild pretty heavily. Last year, I realized I'd turned it into sort of a bullet list of terms, and it was just not doing anybody any good. But this year, we have a good solid set of material that I think everyone can get their heads wrapped around. So you can see the topics that I've highlighted here. I suppose it's been apparent to everybody, but if you're trying to figure out what's on the quiz, the things that make it into the overview slide are generally a pretty good cue for that. So if you don't know what any of those terms mean, it's a good time to find out what those mean. All right, now we've been talking an awful lot about transcriptomes and genomes with our big diagram of all these different workflows. We're now moving into kind of this big jumble over at the left. We see that we have tandem mass spectrometry data entering at the top. Let me grab my laser pointer. We have tandem mass spectrometry data entering at the top, and we use database search identification, which also actually feeds off of protein sequences that we provide, produces identified spectra, which we then have to filter in order to keep just those that have solid hits, and we need to infer proteins from them. And in some cases, we go on to do differentiation to ask which proteins changed between these cohorts. We also, however, need to be thinking about data from crystallography, or sometimes from NMR or electron microscopy. Those can also be used for solving structures. And once you have a protein structure, there are all kinds of interesting things you can do with it. So this is kind of an overview of where we're going with this lecture. First off, though, it's useful, I think, to talk about proteomics. Defining proteomics can be a bit of a mess. There are people who say it's the protein equivalent of genomics, or the protein equivalent of transcriptomics, and yet there are other people who will lump it in there alongside microarrays and say it's functional genomics. We're trying to figure out which bits of the genome are functioning at a given time. Well, those are all kind of a bit distant from what we can accomplish with proteomics. So I kind of like this definition from the Human Proteomics Organization. You see HUPO down here. This is where you can find information about that. So proteomics focuses on the identification, localization, and functional analysis of the protein makeup of the cell. The proteins present in a cell, together with their function, subcellular location, and perhaps even structure, may change dramatically with the organism and the conditions faced by their host cells, including age, checkpoint in the cell cycle, and external and internal signaling events. Well, we went through kind of a laundry list at the start of the last lecture about all the different things that can make gene expression different. And we see the same right here. There are lots of different circumstances that can change what the protein complement of the cell is. In fact, there are some proteins that are completely inert as produced until a signal arrives, which causes them to be phosphorylated, and suddenly all sorts of things have swung into action. So even having a protein manufactured and ready to go doesn't activate it until the right modification takes place. So I want to change us also in how we're thinking about what measurements go into this. Frequently when you're working with microarrays or you're working with a sequencer, you are measuring lots and lots of different molecules all at the same time. We saw our little video of the flow cell for an Illumina sequencer, and we saw that each of these little patches of light represented a different sequence that was building out base by base. In proteomics, we're dealing with an almost entirely serial technology. So instead of measuring thousands, millions of different reads at the same time, we are measuring at one given moment for a mass spectrometer another tandem mass spectrum, and that tandem mass spectrum represents one peptide. So you can imagine that this instrument is just firing off tandem mass spectrum after tandem mass spectrum, many of them every second. And those tandem mass spectra are a high-speed serial collection of data for this sample. You're not getting all peptides measured at once in this technology. So when I say serial versus parallel, I mean serially, I'm going to measure this, and then I'm going to measure that, and then I'm going to measure the other. With parallel measurement, we're measuring lots of things at once. So, what is a mass spectrometer? It's a kind of bewildering thing to ask, frankly. You might go visit the Proteomics laboratory over in the clinical building, for example, and you would see a nice plastic box, about tall off the ground, and it has little lights blinking on it, and this technician says, what is this thing doing? And that's great, but internally, you don't have much sense of what's going on. So let's start by saying mass spectrometers can only measure things that are ions. Ions. So when I talk about when I use the IONS word, it means I'm talking about something that we can measure. If something is not an ion, we can't measure it. Therefore, there's a lot of emphasis on these methods that lead to our ability to measure in a mass spectrometer different biological compounds. Two of the most significant of these are called MALDI and ESI. So MALDI stands for matrix-assisted laser desorption ionization. It means that we're firing a laser at a sample that's been buried in matrix, we get a big plume of ions, and from those we can start our analysis. So it's kind of a pulse technique. The other one is electrospray ionization. I think of that sort of like a cattle prod on a water hose. You got this pile of voltage that you're applying to all the ions that are coming out of typically liquid chromatography, which creates this spray. Both of those are called ion sources. We are ionizing some biological material. We're producing either a flow or a pulse of ions. Those in turn can be drawn into the instrument, into the vacuum environment of the inside to be analyzed. Now I've already used the word analyze a few bazillion times, and sure enough, the next part of the mass spectrometer is a mass analyzer. There are a whole slew of these. Does everyone know how a TV works? One of the first types of mass specs we used was called a sector, and it bears a little resemblance to your TV. So this is before we had all the flat screens. Does anyone remember the old tube TVs? Good, I'm glad people remember the tube TVs. All right, I don't feel old just yet. So we have at the back of a TV an electron gun, right? But that gun doesn't pivot around like that. It points in exactly one location. Now, near that gun, we have some big magnets. And these magnets are able to exert a force on that beam of electrons. And they can position where that beam of electrons is going very precisely. So there is some distance between this electron gun and its focusing equipment and a screen in front that's covered with phosphors. If you are pointing this beam of electrons at this particular phosphor, you get red light. If you hit the one next to it, you get green light. If you hit the one next to that, you get blue light. We've heard this story before. This combination of colors, quite close together, produces the color that you see on a tube TV. But what I want you to pay attention to is the fact that we are redirecting ions by magnetic fields. In the same way that your old school TV directed that electron beam to particular locations, we can apply a magnetic field to a stream of ions, and they will change direction in relationship to how much mass to charge that ion has. So if you have 1,000 Daltons and it's doubly charged, it's going to be a 500 m over z ion. M over z, mass to charge ratio. That shows up all over the place in mass spectrometry, so indelibly bury in your mind right now, m over z is the property by which mass spectrometers manipulate ions. Okay, so the old school sector mass analyzers would use this electrical field to bend the path of ions, and by the amount of deflection in its path, you could discern what its mass to charge ratio was. That's a very old school method, but I think it's one everybody can get their heads wrapped around. Today's mass analyzers are considerably more complex in what they can accomplish. The two that I spend the most time thinking about are Orbitrap mass analyzers and ion traps. I'm not going to get into how they differ in their operation, but you should know that mass spectrometers can differ significantly in how precisely they can manipulate a set of ions. Some of them are trivially able to tell apart the isotopes of an intact protein at charge plus 20. That seems incredibly like a tiny little difference for these instruments to be able to tell apart, but it is in fact possible with Fourier transform mass analyzers. When I say Fourier transform, a neuron should have fired in the back of your mind because we've talked about FT. Fourier transform is about being able to measure frequencies. So we see that technique shows up again. So that's brilliant. We have an ion source, Moldy or electrospray. We have a mass analyzer that allows us to manipulate the ions by their mass to charge ratios. And finally, we have a detector. One of the simplest kinds to understand is an electron multiplier or a conversion dynode. I've seen that term used also. Essentially, you have some ion come barreling out of nowhere and hit this electrode. Well, that is an energetic collision, and that energy is translated into the release of electrons. Maybe just a couple electrons. But those electrons arrive at the next part of the detector, and they kick off a couple more electrons. Those 4 electrons now kick off 8 electrons in the next stage. And eventually, by this exponential increase in voltage, we are able to get a measurable current. So most detectors are going to produce a measure of how much electrical current was caused by the arrival of this packet of ions. That's a destructive detector. It's not the only kind, but I think it's one that's pretty easy for everyone to get their heads wrapped around. The arrival of ions kicks off a voltage. Okay, so what does a proteomics experiment look like? Yeah, that tea is really cold. Okay, so typically, biologists don't care tremendously about peptides, but they do care about intact proteins. So at the start, I see that we have a protein mixture. If I blow up a bunch of tissue, how many proteins are there in a given protein tissue, a given proteinaceous tissue? Maybe it's muscle. Maybe it's a cell culture. Maybe it's an immunoprecipitation of a protein. There are lots of different kinds of samples. But if I'm starting with something like a cell line, how many proteins would you imagine are there? Is it 100? Is it 1,000? Is it 10,000? Is it 100,000? Anyone want to give me an order of magnitude? I think most people feel that a large part of our genome of 20,000 genes are active in a given tissue, but they're not equally active. Some are hugely active and some are not so much. You may have only a dozen copies per cell of a particular protein. And if that's true, you're not going to see very much of it in a mass spectrometry experiment, because those proteins that are present at high concentrations are likely to dwarf any other signal you might see. So just for a round figure, let's say that from a human tissue sample, I have about 10,000 identifiable proteins. That's a round number, but it's a fine place to start. So if I've got these 10,000 proteins, it doesn't necessarily make a lot of sense that in the very next step, I would digest all of those up. But that's exactly what we do in most shotgun proteomics experiments. I've called this discovery proteomics. Another nickname for it is shotgun proteomics. So you can imagine that these 10,000 proteins have been digested with trypsin. Trypsin doesn't just cut randomly. Where does trypsin cut? arginine and? Nope. You got the first one. Lysine. Yes, we have two basic amino acids, arginine and lysine. So having started with a mixture of 10,000 proteins, we've digested them up, and now we've got 100,000 peptides. Well, what a mess is that? One of the things that we're going to have to do to deal with this absurd level of complexity is to fractionate that peptide mixture. This, by the way, is probably the #1 thing missing in South African proteomics at this point. Mass spectrometry experiments are kind of expensive here, and as a result, almost no one uses fractionation in this city or in this country. So here we see that we now subdivide this mixture of 100,000 peptides into fractions. And these fractions overlap a little bit with each other in which peptides they contain, but not tremendously. Each of these peptide fractions can then be subjected to a separation, and the most common separation that we see in proteomics is liquid chromatography. Specifically, reversed phase liquid chromatography, where we have beads that are absolutely covered with C18, long chains of carbon. And hydrophobic peptides love that. That's brilliant. But if you've got a peptide that's covered in arginines and acidic and basic residues, well, that's not going to stick so very well to these materials. So they're differentially adhering to the beads in these columns. We then gradually change how hydrophobic that environment is until all of the peptides have been knocked off of these columns. As we change that hydrophobicity, we're capturing that some of these peptides are flowing off of that liquid chromatography column. And that's when we Zorch them. I mentioned electrospray just a moment ago. They hit this voltage difference of like 300 volts, and blammo, they create this plume of ions at the end of the column. That plume of ions is then going to get measured two different ways. We are going to create mass spectra, and we're also going to create tandem mass spectra. That is the difference I want everybody to understand. So we have the ability to do a high-resolution mass spectrum in instruments like the Orbitraps over here that produces us a very, very accurate picture of what ions are present at what mass-to-charge values. I mentioned ions. Now, what are the molecular character of these ions? Well, they're the same peptides that we produced all the way back here. Peptides can ionize through electrospray. So as peptides are released from the column, they come out, get hit with this huge voltage. Now we've got intact peptide ions, and this mass spectrum is effectively the list of all of the mass-to-charge values to very high precision for each of these peptides. Now, if you have a very accurate mass for some ion, let's say it's 1,284.638 Daltons, is that enough to know what its peptide sequence is? People are saying no. There are people in the mass spectrometry community who would say yes, but I tend to doubt that, that no matter how precisely we know that, we would like some sort of confirmation that not just the mass, but that the fragments producible from that peptide both agree with a claimed identity. So for now, we're going to talk about the mainstream approach here, which is that we don't want to work just off of an intact peptide ion mass-to-charge value to determine the sequence. We also want to see its guts. And for that, we're going to use tandem mass spectrometry. So tandem mass spectrometry also produces a mass spectrum, but in this case, the ions that you measure in your tandem mass spectrum are the fragments of a particular peptide. Except in some new methods that you're doing, but that's your project. So we have a mass spectrum that shows us the list of peptide ions that we can measure. We have a collection of tandem mass spectra that represent the fragments of an exploded peptide. Now, people get a little confused frequently about all these different disruptions. So let's start with the experience of the protein. The protein was just minding its business when some researcher grabbed it and denatured it and reduced it. Proteins, after all, have this complex three-dimensional structure. We're going to spend plenty of time talking about that. So that protein structure means some parts of the protein are accessible and some parts are not. And we really want this protein to be all stretched out so we can do awful things to it. So we're going to denature it, typically with a bunch of urea that causes that structure to come apart. And we're going to reduce the disulfide bonds. Think of these disulfides formed between pairs of cysteine in the structure as safety pins. You're not going to be able to get this thing stretched out if it's got all these little safety pins all over it. So we need to get rid of those as well. We can reduce them. So now, where we started with some nicely formed proteins, we have more or less stretched out linear polymers of amino acids. We can then use an enzyme to chop those. I mentioned trypsin already, cuts at arginine and lysine. Trypsin is going to generate this set of peptides. Are any of these first steps for the first two bullets here performed inside the mass spectrometer? Nope, this is all carried out with your hands. Very frequently you'll be reading in a proteomics article that it was left for, the trypsin was allowed to digest these proteins for a period of 8 hours. When you read that, you should think to yourself, there's no one sitting there with a stopwatch for 8 hours. Instead, this person set the proteins in trypsin and went home for the night. When they came in the next morning, they had peptides. So this carefully covered 8-hour protocol is very frequently somebody left them to soak overnight. So now these steps have all been carried out, and now we have our mixture of peptides. It's only in peptide form that these peptides reach the mass spectrometer. There are also branches of proteomics working with intact proteins. There are some really cool people working in that space. Neil Kelleher coming to mind. But the bulk of proteomics that you see out there has people working with digested peptides from intact proteins. So it's only this last step of disruption, the production of fragment ions starting from a peptide ion, that takes place inside the mass spectrometer. Okay. So I've already emphasized this, but I'll cover it one last time. In shotgun proteomics, we typically have two different kinds of scans. They look very similar. This is a mass spectrum. It's an intensity scale. Intensity is kind of our proxy for how much of this particular ion there is. And across the x-axis, we have the mass to charge scale. A tandem mass spectrum is on the same axis. It's still intensity, it's still mass to charge. But this time, the pieces that we see are all pieces that were created from blowing apart some peptide ion. In fact, we have this little green stripe here to remind us. That green stripe in this case says the ion that we blew apart to create these fragments had a mass to charge value of 898. Okay? So these fragments belong to something that had this mass to charge value. If you're really speedy, you might have glanced at that tandem mass spectrum and said, now hold on a second, something doesn't really add up. You had an ion that had this mass to charge, and you blew it up. But some of your fragments are bigger than that. How could you blow up something with mass to charge x and get something that has a higher than x m over z? The answer is that sometimes when you blow things apart, the fragments don't take all the protons with them. Here we had an intact peptide that was carrying 2 protons. When we blew it apart, some of the fragments had just 1 proton on them. So instead of being divided by 2 to produce their mass to charge values, you divide it only by 1. So you get a larger m over z value from a lower m over z precursor because of the charge difference. All right, so the mass spectrum represents the peptides flowing off the liquid chromatography right now. The tandem mass spectrum represents the fragments from a particular peptide. How does that look? I note that you could have a peptide of arbitrary sequence here, maybe 25 amino acids. That would be pretty big. But this one is just 10. This is a well-behaved peptide, let's just say. This is 10 amino acids, therefore there are how many internal peptide bonds? Peptide bonds link what? Amino acids, good, all right, good place. We have 10 amino acids in the peptide, therefore we have how many peptide bonds holding them together? What? 9, that's good. We have another fence post problem. Fence post problems show up all over the place in informatics. Sometimes you have to remember you're off by 1. So we have 10 different amino acids that are connected by 9 peptide bonds. Good catch. So let us then imagine that this fourth, 1, 2, 3, 4, the fourth peptide bond is going to be cleaved in a collision-induced dissociation shattering. I'm not going to get into all the magic of how CID happens. We blow them apart. That's good enough for now. So we have this breakage of the fourth peptide bond. We get two kinds of ions from that. You might have a proton that leaves with TSII. And because this is an N-terminal fragment, the left side of the sequence, and it contains 1, 2, 3, 4 amino acids, we call that a B4 ion. And so we see that right here we have a mass of 415.2 that represents those 4 amino acids leaving. Now, if I were going to be really pedantic, I would say this has a mass of X, this has a mass of Y. We could go across this. So we can add together all of those amino acid masses, add 1 for the ionizing proton, and we would come up with the number 415.2. Grand. Similarly, we have another 6 amino acids here. Now, I like to joke that we call this the before ion, therefore we can call this the after ion. But that's okay, I'm not going to go there. Right now, we could say that we have these 6 amino acids all in one fragment ion containing the C-terminus of the peptide, and it carries the name Y6 because it has those 6 amino acids in it. Where did A and C and X and Z go? There's a story behind it, but I won't get into it. Blame Ropesdorf and Fulman. So, we now have these fragments, and we see that the fragments we observe from the sequence are indicative of its sequence. Now, who would feel comfortable reading the sequence from these fragments? I'll give you a clue how you do that. Look, if you had a break here, you would create a B3 ion and a Y7 ion. Does everyone see that? Because the left contains 3 amino acids, the right contains 7. If you were to do that, you would get a Y ion that's how much more massive than the next smallest one? The only difference between them is that the Y7 contains an extra isoleucine versus the other one. If we know that this one's mass is 572, we can add the mass of isoleucine to it, which is 113. I'm going to correct that. 113. It's the most common amino acid mass because isoleucine and leucine are very common. So you could add 113 to this and get what? 572 plus 113 is? Thank you. You're sitting right up close, so you can see it right there. That's brilliant. So this is the Y6 ion. This is the Y7 ion. So you can see that as an ensemble, all of these fragment ions together give us a way to confirm that this sequence is represented by this spectrum. Now, I said that a particular way. I feel pretty comfortable about my ability to say this sequence is or is not this spectrum. But I find it a lot harder to look at this spectrum and just read the sequence off of that. I spent a whole semester in a course that attempted to do just that, and I have to say, it's friggin' hard. So we would like to have some software to help us with that. In fact, we'll take a whole suite of software to do that. We started with proteins at the bench. You diligently digested them by leaving the basotropin overnight. Then we collected a bunch of tandem mass spectra, each tandem mass spectrum representing the dying screens of one peptide. Now, we want to be able to put together the list of peptide identifications. We want to return to a set of sequences based on the spectra that we have collected. Then we want to figure out which of those sequences are actually reliable. And finally, we will infer a set of proteins that explain all those peptides we saw. That's the total pipeline. Let's spend a little time looking at the engine behind it all. Database search engines were first introduced by the Yates lab in 1994. Disclosure here, John Yates was my PhD advisor. I had no idea what I was getting into when I stepped into his laboratory, but 2 years ago, he had just published the most momentous paper in proteomics history. My opinion, it's great. You don't have to repeat that. So, we see then that 1994 was this benchmark year. Our ability to transform piles of tandem mass spectra into piles of sequences became a real deal. How did we do that? I already said, trying to just read sequences off of the spectra hurts. It's a really painful, awful experience, and you sit there adding amino acids and subtracting amino acids and looking for water losses. It's freakishly terrible. So, let's use software. The software is going to start by looking up the answer in the back of the book. I already mentioned FASTA databases in a lot of contexts. FASTA databases are just long lists of protein sequences. So with these protein sequences, we can emulate in silico, in software, to say what peptides should occur if I'm given these proteins. All right, so maybe we have a 70,000 protein database for humans representing all the proteins we know how to make. In silico, we can project what peptides should they produce. From that, we can take into account a set of post-translational modifications that might be riding on top. After all, if you have something like a phosphorylation sitting on a serine, it's going to change the mass of the peptide and of the individual amino acid. So those are problematic. We're going to leave those out of much of this discussion because they're a pain. Next, the peptide mass filter. This peptide mass filter was the thing that made sequest work. And ironically, there's a whole series of publications out there right now talking about how we don't actually need the peptide mass filter now that we can measure ions so accurately. So this is kind of a transitional moment for it. The peptide mass filter is the key to our knowing which sequences should get compared to which spectra, to which tandem mass spectra. After all, if you measured, well, just a moment ago we were looking at a tandem mass spectrum, well, right here. Remember we had our precursor measurement? That precursor ion is the thing we blew up to create these fragments. So I know that this one has a mass to charge value of 898.92. This is where we're going to put that to work. At the peptide mass filter, we're going to say if your sequence mass is too far from 892 point, anyone remember the number? I don't. If you're not close enough to that number, we have the right to ignore you. Only those peptide sequences that are very close in mass to what we measured for this peptide will be considered for comparison to that tandem mass spectrum. So the peptide mass filter saves us a lot of work. We now have some set of peptides that were close enough in the measured mass to be compared to that spectrum. So what we're going to do then is predict what the spectrum should look like if it's peptide 1, what it should look like if it's peptide 2, what it should look like if it's peptide 3. And out of all of these images of spectra that we're generating in silico, we're going to figure out which one is most highly resembling the spectrum that we've seen. Which means that we need some way of scoring those matches to say, this one looked very much like what we saw, this one looks very little like what we saw. To re-emphasize, the peptide mass filter is valuable for restricting the set of peptides that should be compared to each spectrum. So if you imagine that you have a measurement at 493.7 or so, if you know the accuracy with which you've measured that to a high degree of precision, you can rule out huge numbers of peptides from ever being compared to a given spectrum. Some people have argued that you can use this mass filter all the way down to saying you have only 20 different sequences to compare to a spectrum. Typically that's in pretty odd circumstances. I'm not really going to go into that. Generally speaking, you will see that if people have measured this in an ion trap, which is kind of an old school instrument, but which was hugely popular when I was in grad school, you would use something like 1.25 m over z. That may not seem like a really big gap, but in fact it's huge. If you digest up the human proteome and look for all peptide sequences that fall within 1.25 m over z of something, you might well have tens of thousands of peptide sequences you have to compare. So that's not great. But today, many people are using time-of-flight instruments, which may be accurate within better than 100 parts per million. Parts per million. So divide the difference in mass between what you observed and what you expected, and multiply and divide that by a million to get a sense of how accurate that is. And instruments like the Orbitrap fusion that we have over in the Proteomics laboratory are typically correct on the mass within 10 parts per million. So you can imagine that if you know the mass of a peptide in Daltons, that's like being accurate within, say, 0.002. It's incredibly good. Very, very high mass accuracy. So things have gotten a whole lot better since the bad old days when we only had low resolution measurements, low mass accuracy measurements for our peptides. Now, how do you score the set of spectra that you have against the set of spectra for the peptides that we're trying to compare to them? That is kind of an odd question. And lots of people have put together lots of interesting answers on the subject. At present, there have been something like 30 different peptide identification search engines published. If you just count those, that were published in peer-reviewed literature. Something like 30 of them. I've already contributed a couple in my own right. So this field is absolutely littered with database search engines. I'm going to bring us back, though, to sequest, which was published all the way back in '94. sequest uses something called cross-correlation. Now this math, I'm sure, is very exciting to you after lunch. So let's try something a little simpler. I have this spectrum that I have observed in my instrument. I have this spectrum that I have predicted given a particular peptide sequence. So you can imagine that if I lay those right on top of each other, I may find that I have these sticks pointing up at exactly the same M over Z's. But I'm also going to think about it if I offset them very slightly. I'm going to offset this one so that the peak at 1,000 that I observed is now aligned with the peak that I observed at 1,001 in my model. I'm going to keep scooting it just a little bit at each location. So this is the same thing as a cross-correlation with an offset value. So you see these little taus. These represent how far these things are apart. This is what a right answer looks like on something like that. When you look at tau 0, you're aligning the peaks that you model at 500 with the peak that you saw at 500. So in a correct identification, you have this spike right at the 0 position and relatively low values outside that. And when we try to score how good a hit this is, we compare the height at 0 with kind of the average of this noise right in here. So you might think to yourself, well, I don't really know how to interpret that number. But in mechanical engineering, cross-correlation has been found to be extremely useful in a wide variety of circumstances. So the mechanical engineer who was coding sequest, Jimmy Yang, agreed with John Yates, who was his boss, that this was a pretty good way to do scoring. As a result, sequest was able to launch, and lots of people who bought thermo instruments got a copy of sequest along with it. That's brilliant, and frankly, it helped pay for Jimmy's house. But a lot of people complained that sequest is kind of slow. As you might imagine, running all this math is actually kind of wasteful of computing time. Right? Computing cross-correlations for thousands of spectra is just, it's a messy process. Jimmy Yang was looking through his code in 2008. How many years later? It was 14 years. 14 years later, he realized that there was a place he could have simplified his math in the original sequest, and he published it in this 2008 paper. That little change reduces the amount of time required to run sequest by an order of magnitude. 10 times faster for one little code change. You have to imagine how many computers burned how much extra time because he didn't spot this little math simplification. It's frustrating, but these things happen. No one's perfect. Now, at this point, I would love to pull out the big jar of marbles that I keep in my office for just this discussion, but for now, we'll just imagine we have marbles instead. Statisticians have also taken a look at this question of how do you evaluate whether these peaks you've predicted for a sequence are a good explanation for the spectrum. And so you can imagine then that the spectrum is no longer a spectrum, now it's a jar of marbles. Each marble has a mass to charge value written on it. At some locations where you could have had a peak, you didn't see one. So you just write down you just write the mass to charge value on a white marble and chuck it in the jar. Some places you did find a peak. So maybe at 876 we did have a peak. So we'll write that on a black marble and put that in the jar. So when you shake it all up, this jar has a pile of white marbles, places where you saw nothing, and a bunch of black marbles that reflect places that you did. Now, we have a population from a statistical point of view. We are now going to draw out the marble corresponding to every mass to charge value at which we should have a peak if this were the correct sequence. So pull that out, pull that out, pull that out, pull that out, pull that out. In this case, I have a jar of marbles that has 1,000 different locations where I could have peaks. 100 of them are places where I actually saw a peak. Now I'm going to pull out 20 marbles that all correspond to the locations where I thought a fragment ion should be. And lo and behold, when I look in my hand, I have 15 black marbles and 5 white marbles. Weird, huh? So just from a random perspective, if I'm just looking at the jar that I started with, what's the probability that the first marble I draw out of that jar would be black? 1 out of 10, exactly. There are 1,000 marbles in the jar, 100 of them are black. So our chance of drawing a black marble by random chance in our first draw is 1 in 10. Good, okay. So what's the probability that 15 marbles that I drew out of 20 would be black in a jar that was only 1/10 black marbles? You can compute this in the hypergeometric distribution. I know you're really excited about that math today, but that's fine. Let us just say that each of these represents a combination, and out the other end pops this very, very low number. If I drew 20 marbles out of this jar, what number should have been black by random chance alone? 2. Right. In general, we would have expected that something like 1 in 10 of all these marbles, to some approximation, would be black. Therefore, we would have expected 2 out of 20 to be black. Instead, 15 of them were. So that's an astonishingly unlikely thing to happen by random chance alone. We can compute that probability, and that's where it falls out. So 3.6 times 10 to the negative 12th is a minuscule number. We can then use these probabilities to score each of the matches to a given spectrum. Okay, that's grand. So we have a way now to put a putative sequence on each spectrum. That's grand. But wait, some of them, the software did the best it could, and it gave some score to the best match, but the best match is still pretty bad, right? What if you only matched 3 ions for a sequence, but that was the best you did against this entire database? It's still not very believable. So we needed ways to control PSM error. PSM is a term that shows up a lot in the literature now, so it's a good term to know. So it stands for peptide-spectrum match. It's not enough to have the tandem mass spectrum. I have a peptide sequence that I am claiming is the explanation for why I saw it. But we want to know, are we going to accept it or are we going to reject it? Distribution modeling was one of the answers that came about for this. A fellow named Alexei Nesvizhky, who's a good friend of mine actually, created a system for modeling true and false distributions for these that allows us to put a different probability of error on each PSM. A probability of error on each PSM. That probably reminds you of something. That's right, back in the genomic sequencing work, we were talking about Phred scores as a probability of error for each base call. So the corresponding way to do that in the proteomics world is to come up with a probability for each PSM. The math behind it is too complex for a lecture that has to cover these many topics. So for now, I'm going to bypass that. But I will talk to you about target-decoy search. This is a method that people use all the time in proteomics, and it's very much worth your knowing. Imagine that I have sorted all of the PSMs from a given experiment from best score to worst score. All right, so these ones down at the floor, these are PSMs that matched maybe a couple fragment ions, and that was it. They're lousy. The ones touching my fingertips, though, they're brilliant. Brilliant. 20 matched peaks. Very, very low signal to noise, blah, 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 blah. A very high signal to noise, my bad. This is grand. So somewhere from brilliance to trash, I have to draw a line. I'm going to try to set a threshold that says this set of peptide spectrum matches are believable. All right? So for that, I am going to allow this line to move up and down, and in that, I'm going to try to measure what my false discovery rate is going to be. We've talked about false discovery rate already, Benjamini, Hochberg, etc. This is a way to do this in the context of proteomics. So what we're going to do is estimate what our false discovery rate is for a given location. Well, how do we do that? We're going to start by the fact that we're going to add sequences to our search space, to the original sequence database that we started with, that we know are bogus. How do we do that? Well, the simplest way to do that is for every sequence you put in there that's really from your organism, just flip it. N-terminus for C-terminus. Give the sequence in reverse order. Generally speaking, biology doesn't produce a whole lot of palindromic peptides. These are peptides that are the same in either orientation. So you can generally get away with including the reversed sequence of each protein, and anytime you have a spectrum that hits a reversed sequence as its best hit, you know that thing is false. You absolutely know it, because that sequence doesn't exist in nature. Therefore, if it hits that sequence and that's the best it can do, you can be pretty darn sure it is a bogus hit. So, remember our stack. We have our very good spectra, our really trashy spectra. If I throw a line across some point in this stack, I can count how many of the hits above that line are sequences that might possibly be true, and what what number of them are certainly false. Because here's the magic. If the software could not figure out what the answer for this spectrum was, if this spectrum is falsely identified, it has an equally good chance of matching to a reversed sequence that we know is a decoy, as it does to matching to a sequence that might possibly be true. So think of it this way. Every time we have a known false hit above our threshold, we suspect that it's got a secret partner somewhere among the possibly true sequences that's also false. So when we compute our false discovery rate to say what fraction of the IDs above my threshold are real, we're going to double the number of decoys, reversed proteins, reversed PSMs that we hit, and divide that by the total number above our threshold. So what we're doing is controlling the rate at which we can accumulate false discovery peptide spectrum matches. Typically, you will see people controlling that rate to 2% or lower false discoveries. All right. Now, it might seem trivial to determine what proteins you've identified if you've got a whole set of peptides. It might seem trivial. It's actually quite messy. So, we've already talked a little bit about orthologs. We've talked enough about sequence comparison. We've also talked about the fact that paralogs can exist. Paralogs being places where gene duplication over evolutionary time have given us lots of copies of a particular gene. So, orthologs and paralogs are there. If you are working with a sequence database that contains many, many species, you will find that orthology is a problem. If you find a peptide for human actin, it might mistakenly make you think that orangutan actin is present, or chimpanzee actin is present. So orthology creates a problem in that you might mistakenly think you've identified a peptide from an altogether different species than your sample represents. That's awkward. Next, you might have a case where paralogs are present. You could imagine a scenario in which a peptide is found in multiple isoforms, multiple transcripts, all from the same gene. As a result, identifying that peptide might make you think, well, I've got all of these different isoforms observed, or I have peptides from all of these different genes that happen to be a paralog set. That too is awkward. So we need to be rather more careful in claiming that proteins are present. We need to have peptides, ideally, that are specific to the sequences that we've observed. Now, I love talking about parsimony. This is a principle that we really don't talk about enough in biomedicine. But I'm going to be really brief about it, simply because it's already 3:02, and I don't want to run us late. So, parsimony means that we need to be very stingy when we're doling out explanations from our data. The principle of parsimony says that if you have the peptides that would allow you to claim 15 different proteins, but only 2 of those proteins would have all the explanatory power of the full set, that you limit your explanation to just the 2 proteins rather than the full set. You don't want to go beyond the necessary to explain your data. This comes from a fellow named William of Ockham, and we frequently say that he said, plurality ought never be posed without necessity, which is to say your explanation should never have more parts than the evidence you're basing it upon, more or less. Now, in reality, he didn't really say that because our language sounded really bloody different back then. But he did write in the 14th century, pluralitas non exponenda sine necessitate. I'm sorry, that probably sounds more like Italian than Latin, but he was arguing that your explanation should never step beyond what your data can support. So when we see multiple possible explanations for the peptides we see, we should always choose the one that invokes the fewest proteins to explain the peptide evidence we have. What does that look like? This is called a bipartite graph because it's got proteins and it's got peptides, and all of the links run between the two. So here we have table 1X isoform B human that could explain these two peptides, and it can explain that one peptide, and it can explain that one peptide. You can see what all these arrows mean. This was a complex sample that was a mouse xenograft. Has everyone heard of xenograft? Maybe not everyone. Okay, so a xenograft in this case meant we were studying breast cancer. We had human tumor cells injected into mice. Therefore, we had a tumor growing in the mouse guts, which means that the proteins we pulled from those xenografts could be from mouse, or they could be from human. So we have both human and mouse variants of proteins, all pointing to these peptides. And we need to make a decision about which proteins we are going to claim. Now, I've carefully drawn a little double box around this one and around this one. In this case, it might appear that these 7 proteins can all be claimed. But the reality is, if you invoke this protein and this one, you've already explained the presence of all of those peptides. So we must be parsimonious when we're explaining which proteins we found, and limit ourselves to the smallest list that can possibly explain the data we've observed. Okay, so that's parsimony. Does it in practice have a big effect? Well, I mentioned the problem with orthologs a moment ago. What happens if you use a database like UniProt or SwissProt for your identifications? Now, about the time we did this study, there were 264,000 proteins in SwissProt. So we just used that. Here's SwissProt. This is the SwissProt multi-species. For part of this, we yanked out only the human proteins from SwissProt. And in this case, we used a database that was trying to be very complete to include all transcripts known for humans. Starting with our data, which were a known mixture, a defined mixture of 49 human proteins, we asked, how many proteins can we claim were present in these samples from all these different database searches? You can see that if you just say, if I saw a peptide for it, it's part of my claim, you end up with a really long list of proteins in each of these cases. We know that the sample contained 49 proteins, and here, our software is trying to tell us that there's upwards of 400 proteins present. Well, that's ridiculous. So what we found is that if you start by grouping indiscernible proteins, this is to say when you have 2 different proteins that have equally good, exactly the same set of peptides observed, you treat them as one protein. You see that the number of proteins claimed takes a huge dive. And if you apply parsimony on top of that, to say, for example, this protein explains peptides A, B, and C, and this other protein only explains peptides A and B, you can then just rule that one out, call it a subset protein, and don't let it cross your mind again. Well, now you have an even smaller list. And even if you start with a massive sequence database, the number of proteins you invoke no longer rises with the size of the number of the size of your sequence database. So it actually has a rather big effect on the number of proteins we claim. Parsimony is considered a necessary element of protein calling by most people now. All right, now I need to take a brief moment to re-liquefy my vocal cords. Are there any questions? Boy, that tastes bad. Okay, let's make an abrupt shift. We've been talking about protein ID. Now we're going to talk about protein structure. I want to advise you again that if you'd really like to talk with somebody with really deep knowledge in this space, I suggest you chat with Colin Kenyon, my next door neighbor. I had to look through the slides with him to make sure I was covering things that were still relevant. So I think at some point, many of you have experienced biochemistry, right? Everyone remembers it? Did anyone take P-Chem, physical chemistry? I wasn't brave enough for that either. Well, I do a lot of my work in analytical chemistry, and I really enjoy biochemistry, so I'm gonna bring us back to that for just a moment. This is a diagram of 4 levels of protein structure. This is always considered good fodder for asking somebody at a master's defense. If they haven't got this stuff completely covered, we'll probably take them down and beat them a little bit with sticks. Anyway, we start with the fact that proteins comprise amino acid sequences. And those amino acid sequences are held in place with peptide bonds. So we call this the first level of structure for proteins. From there, we frequently see large numbers of hydrogen bonds forming in these localized areas, and those can produce alpha helices and beta sheets. Here's a helix, here's a sheet. In tertiary structure, we frequently, are we still counting disulfide bonds as tertiary structure? I think we still do. All right, so you can imagine then that we've got all of these folds, all of these helices and sheets. Those may need to be fixed in position by a few of these disulfide bonds between cysteines. And you may in fact see that an individual polypeptide is not all that you need to make a protein. You may need to make a dimer, for example, a homodimer if it's the same polypeptide sequence being brought together, or a heterodimer if you've got two different proteins that act together. And you may have cofactors that also show up, things like heme. They're not part of an amino acid sequence, but they're certainly quite necessary to the function of a protein. So when we speak of these four levels of structure, we want to be able to understand the underpinnings of how this protein adopts a particular structure, because as we all know, structure implies some sort of function. So this structural biology piece is our number one link to understanding how a given protein adopts a function. Now, I want to point to this little article from 2003. It's kind of a fascinating little look. Did you know that we had our first X-ray crystal structure for Pepsin, if I recall correctly, in the 1930s? And yet it wasn't until the late 1940s and I think 50s that Linus Pauling was able to set us on the right course for understanding the nature of alpha helices and beta sheets. So all of the theoretical understanding of these sequences was growing as our experimental data began rolling out. Something else I didn't know was that a professor from Howard University, an African American in the United States, began working with Linus Pauling and is one of the authors on the very first paper explaining how alpha helices form. So I thought that was great news. Okay, on we go. So it's a nice little biographical look at how we got to protein structure. So what value is it? Why do we care whether we know a protein structure or not? First off, if you know the structure, you can make strong arguments about the function that this protein takes on. If you don't have that, you're going to be in a slightly more challenging space. Detailed structures define protein interfaces. I mentioned homodimers and heterodimers. Well, understanding how they interact requires a knowledge of what structures they take on. If they don't have faces that are compatible with each other, how could they form a dimer? All right? Structures help us to understand the impacts of sequence variability. This is a big deal. This is a really big deal. Let me give you an example. In a lot of our divisional research, we're concerned about the drug response of tuberculosis. If a bug has a weird variant to one of its genes, it may render that bug resistant to that drug. This is a pretty bad situation. Multiply drug-resistant and extensively drug-resistant TB is on the rampage in South Africa, and we would really like to be able to do something about it. But understanding what effect a given sequence variant has on function really matters, or on drug sensitivity in this case. So mapping a genetic variant to a phenotype remains a very, very big deal. We talked about SIFT and polyphen. If you work with those extensively, I think you will find that you wish they were better at the purposes to which we want to put them. So structures help us to understand the sequence, the impact of sequence variability. And finally, ligand interfaces. Maybe you're trying to figure out how this protein interacts with a drug, if it's a druggable target, for example. Maybe you want to understand how this enzyme catalyzes a particular reaction. For those, having a structure makes a very big difference. All right, now, how do we talk about structure? Long, long ago, before we had decent computers, no, we had computers in 1963, they just weren't fun, right? So I want you, by the way, thank you, Marina, for assembling a dipeptide model for us. Everybody should have toys in his or her office. I'm a firm believer in this. So this is my Molly mod kit. We have our blue objects here. These are representing nitrogens. We have our black objects. These are representing carbons. And we have a couple R groups in here. See this one right here and this one right here? Those are R1 and R2. This is a dipeptide. So what bond am I holding right now? NCC, NCC, okay, what is it? I'm holding the peptide bond. I heard it, thank you. The peptide bond. Is that you, Lisa? Well done. Do you go by Lisa Marie or Lisa? Lisa Marie, well done. All right. So, NCC, NCC. I'm holding a peptide bond. What that means is that I'm sitting between a carbonyl. Everyone remembers what a carbonyl is? C double bond O. That's a C double bond O. And a secondary amine, if I'm not mistaken. Secondary amine, we call that when we've got two carbons attached? Two carbons? Okay, that's fine. So this is a peptide bond. And a thing I want to tell you about peptide bonds is that even though we blow these things apart in mass spectrometry, they're pretty darn robust from a physical chemistry point of view. I learned today that this double bond gets a little less than double bondy, and that sometimes that electron strays over to the nitrogen here. One of the effects of this is that you don't frequently see a peptide bond spinning. You can think of this as a plane, in fact. So we have this very strict non-spinability for this nitrogen connected to the carbonyl. Now, we do have two bonds that can rotate pretty freely. We've got this one right here between the nitrogen and the alpha carbon, and we have the one from the alpha carbon, that's the one with the side chain on it, and the carbonyl after it. Everyone good? So you can think of the peptide bond as a bit of a formal plane that's connected to the next formal plane by two spinning joints. So that alpha carbon can play fast and loose with you. So when you're trying to figure out a backbone structure for the protein, which is not a complete structure, just where does the backbone go in 3 space, you need to think about each amino acid as introducing 2 degrees of freedom, where we can rotate these bonds around, and those are all possible conformations. So a long time ago, someone gave these the name phi and psi. Now I was trying to remember which one is phi and which one is psi, right? So phi is this first one that we have on the screen, and this one is psi. Phi and psi. So you could imagine then that if we were being kind of sloppy, we might say every 5 degrees is a different discrete position. Some of those are good, some of those are bad. When we look at what kind of structures come out of these, we see something that a fellow back in 1963 called the Ramachandran Plot. Surprisingly, his name was Ramachandran. So what we see is that we have different combinations of phi and psi angles. And if you fall in this upper region where you have a high psi angle and a low phi angle, do you call it phi or phi? I could call it phi if that makes anyone happier. Phi? All right. If you've got a low phi angle and a high psi angle, you're probably sitting inside a beta sheet, a pleated sheet. You may, however, have a relatively low psi angle and a relatively low phi angle, and if that's the case, it might be a right-handed alpha helix. It could be that you have a slightly positive phi angle and a slightly positive psi angle. If you do that, then you're probably sitting in a left-handed alpha helix. So this is kind of amazing. If you're looking at a large swath of these phi and psi angles for a region of amino acids that fall in one of these secondary structural formulations, you can plot where those phi and psi angles show up and see that it conforms with these standard rules for understanding secondary structure. Now, I wanted to point to something called steric hindrance. Steric hindrance. Does anyone remember that from biochemistry? All right. So let's just talk about some obvious cases. We have our fixed peptide bond. I'm now going to turn this one like so, and I'm going to turn that one. Oh, sorry, I can't turn that one. Well, I'm going to turn this one around this way. Do you see what I've done? I've made a weird kink in this backbone. Some combinations of phi and psi angles can bring molecules into conflict, can bring atoms into conflict with each other. Something like this does not happen in real life. This is because of steric hindrance. Steric means you've got bodies bumping into each other, so that's not a great combo for this phi and psi angle combo. And as you can see in this figure, he has I'm going to apologize. I just call him Christoph. I hope you don't mind. It's a Polish name. We see that we have this combination of 0 phi and 180 degrees psi, and it brings these two oxygens into close proximity. That sort of stuff just doesn't work very well for physical structures. So in effect, we see that there are these big dead zones where one just doesn't frequently see that combination of phi and psi angles in biological structures. Okay, so that's all fine and good. We like knowing that we can parameterize, we can model what this sequence looks like by giving us, by computing these phi and psi angles. So if I have 200 amino acids in a protein, how many phi and psi angles have I got? It's as many as you have amino acids. And if each of those could assume any of 20 different positions, you would have 20 to the 400th power possible conformations of these sequences. Weird, right? Just because you know the protein sequence does not mean you can immediately leap to its structure. Therefore, we've worked on a lot of chemistry methods that allow us to determine what these protein structures are empirically. So, which of them is best? Which of them is best? You could take the facile perspective and say X-ray crystallography has a lower 1.3 angstrom, we're going to call that angstrom, resolution than does NMR. It's twice as bad. Look at that. 4 to 6 angstroms. That's terrible. And then look at electron microscopy. 10 angstroms. Who would bother using such a thing? But the reality is, there's more to this than resolution. You can only use X-ray crystallography if you can get a pretty crystal out of your protein. And while some proteins crystallize just fine, there are plenty that are troublesome. Imagine, if you will, a membrane-bound receptor. How easy is it going to be to make a crystal out of that? Not so much. Not so much. So X-ray crystallography may be completely off limits for some categories of protein. For something like that, you might be able to use NMR. But look, NMR has these really strong limits on size that you can work with. If you've got some 1,000 amino acid protein, can you use NMR with it? Not so much. So you might be reduced to working with just a small domain of that protein. Electron microscopy has a pretty low resolution. But on the other hand, you can deal with really big stuff. Maybe you can throw an intact hemoglobin through there and get a structural profile of the whole thing. Sometimes 10 angstroms is just what is the best you can do. So keep in mind that our methods to make these measurements empirically is not always of course I'm going to go X-ray crystallography. I want to raise one other issue here, and this is one that people don't often think about. You might think, in looking through the protein data bank, that you found the structure for a protein. You download your PDB file, you visualize it, you stick it on your poster. You all know how to do that from this morning's practical. I'm glad for that. But you should always keep in mind that proteins are dynamic objects. Proteins wiggle around. Solvent waves come flowing through and those vibrations get picked up and it maybe wiggles this loop a little bit. So before you start thinking that this protein always looks just as it appears in its crystal structure, you should remember that there's a little bit of biological variability here. And frequently, when you look at an NMR structure, you get a little better perception of different snapshots of what this particular protein looks like. After all, if you were to stack a bunch of people into a crystal, you're going to deform us a little bit by stacking us up in this way. Proteins are no different. If you jam a bunch of proteins in a tight crystal, they're probably not going to look like they do biologically, where they're just out happily swimming. So, not swimming, but you get the point. All right. Now, I'm going to propose that it's worth our talking about threading, and there's some question about this. Threading, in my view, is one of the best hopes we have for making structural information personalized. Everyone cares about personalized medicine, right? Well, if you have a weird sequence in a particular person and you want to know what its impact is, you could set out trying to crystallize that protein for that person. But if you already have a structure for the wild type sequence, you might find that introducing small perturbations that reflect the ways that this sequence is different than the one that's represented by that structure may get you to an answer a whole lot faster, and it may tell you enough that you know whether or not phenotype is altered by the sequence change. So we call this threading. If you have a related structure for a very similar sequence, you can ask, how would these slight alterations in the structure impact the shape that the structure takes on? Okay. Now there's a server out there that you can use to help you understand threading. That you can give it example changes and see how they work. That's, I think, Threader 3, Threader, something like that, in the United Kingdom. This article from 2015 talks about threading in the context of personalized medicine. Can we use this to understand an individual's particular changes in response to their genotype? All right. I alluded a moment ago to the fact that the number of psi angles and the number of phi angles in a protein is equal to the number of amino acids. So you can easily imagine that if you consider just 20 different positions for each phi and each psi in a protein, you're going to have a huge number of potential ways that this backbone could thread through space. So I want to introduce you at this point to Levinthal's paradox. It's kind of an old concept, but one that I think is very illustrative. So let's start with this weird claim of Levinthal's paradox. It would not be possible in a physically meaningful time to a protein to reach the native functional conformation by a random search of the enormously large number of possible structures. Okay, so if you're thinking about this structure, you can turn this one very slightly and turn it a little more slightly, turn it a little more slightly. Each of those comprises a different conformation for this protein. If you have 200 amino acids and each of them has these 2 bond angles that could be changed through 20 different positions, you have 20 to the 400th power different conformations for this protein. Weird, right? I mean, that's just a massive variability that's available to a rather typical protein. So, some of those positions are disallowed, but you might think that as this polypeptide is released from the ribosome, that it has to sample for a while to figure out what structure is the one that is the lowest surface energy. You might think so. In fact, most protein folding takes place very rapidly. Sometimes it requires a chaperone to help guide it to a proper form, but generally speaking, this process of protein folding is not something that takes an hour or even minutes. So how is it possible that this sequence knows what kind of fold it should go into? So let's ask that question. So how do proteins fold correctly and do it the first time? The introduction of a small energetic bias toward the native state being the ordinary conformation, leads to decreases in folding rates such that they can be compared with experiment. So if there's a slight energy advantage in having this bond at this angle and the next bond angle at that position, if there are very slight energetic differences, they can sort of act like a funnel, more or less, that this protein sequence falls into that native shape by sort of collapsing into it, by having a little bit more impetus towards being in final conformation with each proper fold. Maybe this is a little like grad student days. So imagine that you're a grad student who wears his or her seatbelt. Because you wear your seatbelt, you also think, I could probably drive below the speed limit. I know this is a fantasy, but let's just say. If you wear a seatbelt and drive below the speed limit, you might also brush your teeth every day. If you also brush your teeth every day, you might even try to get proper sleep at night. You see, this is the virtuous cycle. You start acting like a good boy or a good girl, and soon you're living a life of wholesome purity. In this case, we have a polypeptide that starts folding in maybe random directions, but it finds that some of them lead to more and more correct folds until finally you fall into the right fold. So this is an additive process where each little right turn has a bias in favor of it, an energetic bias in favor of it. Okay, let's talk about something kind of frustrating though. Sequence databases are growing like crazy, all the time. There's always some new organism we've sequenced. It's brilliant, wonderful. But even though we're gaining millions of sequences every day, I don't know what the number is, it's a lot, we are gaining structures much, much more slowly. I think I mentioned 1934, 36, I've forgotten already, that a female biologist measured the structure of pepsin, I think it was. By the time we reached 1971, we had enough different structures known from these experiments that the protein data bank got started out at Brookhaven. And we've seen now that several different sites around the world, four major resources, including the RCSB that we were looking at just this morning, these 4 sites have all joined up and compelled their results to be kept in lockstep with each other so that all of us can learn about all of these structures as soon as any of them are known. So what we see is that over time, we have a logarithmic growth, or an exponential growth, in the number of structures that we've got. It's not even close to keeping up with the number of sequences, but I would point out that there are fewer folds in the world than we have sequences. That sequences may be quite different, and yet the structures that they finally adopt are quite similar. So, even though we don't have as many structures as we have sequences, we have an awful lot of sequences in known structures for sequences that are similar enough that we can adapt a new sequence to the fold that we think it's likely to take. We have a lot of software out there, though. A lot of people who've invested a lot of time in trying to predict this structure given just a sequence. What a novel concept, right? Imagine if every protein sequence we could get out of the databanks could be immediately projected into a nearly correct protein structure. That would be brilliant. Think about how much more we would understand about biology. You sequence some new bug, you see a protein that doesn't have an ortholog in any other species, you run it through this software and bang, out comes a protein structure that you can go ahead and use. That would be amazing. But we kind of have to have a scorecard. We need to know how the different toolkits that have been made available compare on sequences that they haven't seen before. And CASP exists exactly for that purpose. So CASP just finished version 12. There's a really moving tribute on the CASP site, you see predictioncenter.org. A woman named Anna, I've dropped a blank on their last name, Tremasov, something like that, who's been part of Prediction Center since its very beginning. She died this year, right after the completion of CASP 12. So participants, the people who are from the outside, are given a sequence, and they are told to send back a structure. So this is an honest-to-God test, and we know the answer, because CASP is always working from structures that have just been finished, but have not been published. It's kind of a nice bit of publicity, right? My unknown structure was used to power this competition. It's a nice little pat on the back. So these are some examples of the protein structures that they have. I remember this morning we were talking about the cartoon structure and little arrowheads that should be showing on the beta sheets. They show up here. And I don't know if they do on this one or not, but in this case they visualized it the same. So every couple of years, someone's structure is adopted as the problem set for this challenge, and these researchers will try to send back their best predictions about it. So one can ask, over time, are we getting better at this? So I would note that In this case, we're looking at 3 different editions of CASP. It's run every 2 years, so essentially we're looking at 6 years of development in the field of protein structure prediction. The precision here is about distant contacts. So we have bits of these polypeptide sequences that come back into contact with themselves at some long distance, more than 24 amino acids apart. That was the rule they used. So if we look at the distant contacts in CASP-10, which was run 4 years ago, we see that the precision they achieved on it, at best, was just over a score of 20. And that's only true for the best person in CASP-10. The others fell off in sort of a smooth line from that, until you get to these guys at the back. They didn't know what they were doing. I'm just casting aspersions on strangers here. Now, look at how we did in CASP-11. Well, we had a little step up. Things got a little better, but you'll see there's just this tiny little pip of green poking above this red line. Essentially, the winner, there are no winners in scientific competitions, but we'll call him a winner. The winner of CASP-11 did a whole lot better than everybody else. That's because all the other green traces are way back behind that mass of winners from CASP-10. So, the people who did use this winning technique in CASP-11 became very influential, and lots of other people took ideas that they had and ran with them. Now, when we look at distant contact precision in CASP-12, we see a tremendous step forward, and it's a crowd of people who've all borrowed these methods. So what was the trick? In this case, machine learning. A lot of people, I think, have heard of a deep learning approach. It's an artificial neural network with lots of layers. People have found that deep learning in particular has been very well adapted to protein structure prediction, and by involving co-evolution data, they were able to really improve distant contact detection in these structures. In this case, 38 different groups took part. Only 36 of them gave back results. This often happens when you do collaborative studies. People get busy. What are you going to do? And then finally, they did a comparison between labs to see who was producing essentially a copycat result. Different labs all using exactly the same software. In that case, they found 32 distinct scores that they wanted to compare. So in total, I think we can say that things are actually getting a lot better in the space of protein structure prediction. Will it be something where we can hand them an arbitrary sequence and boom, out pops your PDB? In some cases, yeah. Will it be a PDB structure that you can trust? Well, your mileage may vary. But if you look at how the winners did, given just sequence, I think we start to feel that this field is maturing rather significantly. I'll tell you this, at Vanderbilt University, they burn a lot of CPU power. Anytime a CASP competition happens, you know there's going to be a couple months where the supercomputer's just like, give me a second. So, how do de novo structure prediction algorithms work? This is the sort of thing that I'm going to describe to you, and I hope you internalize some of it, but I'm not going to expect that you understand this algorithm any more than you might the Burroughs-Wheeler transform. So I'm going to talk about some concepts here and how they relate to this topic, but it's a difficult thing to get. Let's start with an energy landscape. I've kind of made allusions to this before, that certain conformations of a protein backbone are more or less automatically excludable. They bring these two oxygens into close contact, for example. Not too likely to happen. So when we look at a possible solution for a structure, we can already evaluate it on an energy level to say, to what extent is this a viable conformation? We can form a scorecard on that, and typically energy landscape is something that we use as a metric for it. The next is Monte Carlo sampling. Monte Carlo sampling gets used in a lot of contexts, actually. The idea here is that you may not know the ideal combination of these factors. So you try a random starting place, and you see if you get anywhere good. You try another starting place, see if you get anywhere good. You try another starting place, and see if you can get anywhere good. So Monte Carlo sampling lets you look through kind of an undefined space and see if some of these places are hotter than others. Next, hill climbing. So you may find that a you had this starting place and it got an energy score of X. And as you may then try, say, 5 random starting points that are close to that one in space and see how each of those does. And you see that, well, if I go in this direction, my energy score drops. It's a more plausible conformation. So you may direct your efforts, you may sort of steer your sampling in a particular direction because you see that that area is getting you to a lower energy, a better structure. And finally, the idea of a Rotamer Library. This is really about benefiting from what we already know. To build a Rotamer Library, we can build on all of these sequences that we had. If you recall just a couple slides back, we saw that as of 2014, we had in excess of 100,000 protein structures. That's not a very impressive number when we think about how many sequences we've got. But 100,000 structures is an awful lot of institutional knowledge. So what we can do is look at sets of, say, 7 amino acids drawn from known structures and ask, what bond angles did they take? What were the phi and psi angles for each set of 7 amino acids in each structure in that database? So from this, we can build what we call a Rotamer library. For a given 7-letter sequence, we may have multiple sets of measurements from crystal structures that tell us what this 7 amino acid set does conform to. So that's valuable. So I realize that that's waving a lot of hands in the air and giving you a very vague notion of what this type of process looks like. But I hope that it starts to flesh out a little bit what kinds of things these algorithms attempt in order to solve these very big spaces and give us back a concrete answer about conformation. So I've made allusion to energy surfaces. I just wanted to point you to this 1991 Science article. This is the reaction energy for binding a carbon monoxide to myoglobin. Sorry, MB was not ringing a bell for me all of a sudden. So what we see is that through association with an enzyme, these reactions are able to find a reaction that may not be favorable all by itself can become favorable in connection with a protein surface. The very surface of the protein is what actually reduces the energy that is required for a reaction to step forward. So this concept of energy surfaces is not just one that we find in the structural conformation world. It also, just to drag this point through the mud one more time, structure is directly mappable to function. And using the structure, you can learn an awful lot about the energetics of a process that this enzyme or this storage molecule may be trying to accelerate. So in takeaway messages, identification depends upon a database of known protein sequences to define the set of possible peptides. So we start with the idea that we know all proteins that a given species can make. So we're going to use that information to fuel our identification of proteins. We look at peptides from the database, we project what kind of fragments they would produce if this were the spectrum for that peptide, and then we compare that projected spectrum, that projected set of fragments, to the ones that we observed. That's tandem mass spectrometry identification in a nutshell for proteomics. Database search is the strategy almost everybody uses in this field. Controlling error in identified peptides and proteins is critical to reliable proteomics interpretation. We've talked about this in the context of genomics, and I think it bears repeating in proteomics. You may have some protein that you want to be differential in your experiments because your advisor said, I'm sure that little guy is active in this particular response. But we need to be careful that we are controlling the error rate at which we identify these peptides and these proteins. Because if you don't, you end up with a bunch of proteins that are just false discoveries. Everyone in the proteomics field remembers what it was like in the late 1990s, when people were publishing protein lists that were completely unbelievable, literally unbelievable. They were incredible in the sense that they lacked credibility. People don't want to go back there with proteomics. So when you publish proteomic identification data, be ready for skepticism. Be ready to defend your choices for controlling false discovery rate. Okay, protein structures can be measured through X-ray crystallography. We like it, very nice resolution. By NMR, nice, it gives us a dynamic picture of what this protein looks like. Or electron microscopy, cool, we can do really big structures. All of them have their strengths, all of them have their weaknesses. Computational methods in protein structure continue to evolve. Well, that's a kind of throwaway statement. All science continues to evolve. Structures for small proteins can generally be inferred to good approximation from sequence alone. That is not a statement I would have made very happily 10 years ago when I was teaching this subject. So I think that we really have gotten rather better over time because this field continually evaluates itself against an objective scorecard. And that's something that not every field does, so I think we can feel happy about it. All right, it is time for the weekend, so I'm not going to keep you much longer, but I will see you back in here at 9 a.m. on Monday when we will begin looking at the problems of data analysis and quantitative mass spectrometry. Thanks.